everybody. I'm Dr. Deepak Jain, Senior Professor, Department of Medicine, PGIMS Rohtak. Today, the topic of talk is a dissolution technique in the Telmisartan formulation. We all know Telmisartan is an angiotensin receptor blocker, which is approved for hypertension. It binds to the AT1 receptor with a high affinity, causing inhibition of action of angiotensin 2 on vascular smooth muscles, ultimately leading to reduction in arterial blood pressure. The solubility of telmisartan in aqueous solution is strongly pH dependent, with maximum solubility observed at high and low pH. After ingestion, tablet undergoes disintegration and dissolution before being absorbed. Rate and extent to which into the systemic circulation it determines its bioavailability. The solubility and dissolution behavior of any drug is a key determinant of its oral bioavailability and rate limiting step to drug absorption. Poor solubility results in low bioavailability, leads to large inter and intrasubject variation and large variation in a blood drug concentrations under fed conditions and fasted conditions. If the drug does not dissolve readily or cannot penetrate the epithelial membrane, its time at the absorption site, it may be insufficient. Bioavailability tends to highly variable. So it has been seen that absolute bioavailability of telmisartan is about 42% at 40 milligram and 57% at 160. The solubility in aqueous solution is strongly pH dependent, with maximum solubility observed at high and low pH, as already stated. With any oral preparation, drug dissolution profiles may be distinct due to difference in formulations and manufacturing process, but the difference must not compromise products' bioequivalence. It has been observed that different brands of telmisartan available in the market produces different clinical responses. There is a regular check from CDSCO for keeping standard quality. The CDSO, Central Drug Standard Control Organizations, had declared 1,064 drug samples as a standard quality based on analysis and testing done on 1,095 samples last month as a part of random sampling. It means it concluded there were about 31 samples which were detected as not of standard quality, spurious, and misbrand. As I have already mentioned, that because of variable therapeutic responses seen with various brands of telmisartan and unique pharmacokinetic property of telmisartan, comparison of dissolution profile and other parameters of a generic telmisartan brands with that of an innovator brand is required. Various methods are used by manufacturers to overcome the bioavailability and improve dissolution of a poorly water-soluble drug is by formulation of a solid dispersion with hydrophilic polymer, polyvinyl, polyiodine, and other diverse carriers and use of alkalizers. Sodium hydroxide is considered as a strong alkalizer as it provides maximum solubility of telmisartan. Some of the generic and innovator brand contain sodium hydroxide and it creates a microenvironment pH around telmisartan granules and thus enhance solubility of telmisartan intestinal fluid. Unique technology is used by companies wherein micronized telmisartan granules primarily mixed with a suitable alkalizer, sodium hydroxide, an open-label, two-arm, randomized parallel control study conducted to assess the bioavailability of Telista, Lupin's brand, tablet formulation in 18 health male volunteers. The telmisartan peak plasma concentration is achieved within 30 minutes of oral administration and T maximum in 1 hour. The dissolution medium selected is a pH of 7.5 phosphate buffer, which is a media specified for telmisartan tablet as per FDA OGD guidelines. USP require not less than 75% of a tablet amount dissolved within 30 minutes. The dissolution of more than 80% of drug should be released within 1 hour is following the pharmacopoeial specifications. Compared with Micradis, which is the innovator brand, Telista has shown 96.6% of a drug release, comparable with the innovator brand. So just to summarize, Sodium hydroxide is a strong base used to dissolve as the drug is a soluble in a strong base, and alkalinizer used to improve the dissolution profile. And the brand Elista, they have shown that the dissolution media at 75%, this drug is at par with the innovator brand. Development of a dissolution method was validated in accordance with ICH guidelines because providing the method is experimentally solid, yielding precise, accurate, and repeatable results. Thank you very much.